Good evening. Welcome back to the Vancouver Aquarium's Introduction to Marine Life course. Tonight's program, Sub-tidal Marine Organisms. Our instructors tonight are members of the Vancouver Aquarium's Howe Sound team. Established in 1996 to conduct natural history studies and do baseline documentary work on groundfish stocks in Howe Sound. They are the only group in the Strait of Georgia conducting continuous studies of Howe Sound groundfish populations. To date, they have monitored prawn nursery and glass sponge growth as indicators of environmental quality. They have monitored seabed biodiversity for long-term comparisons, documented Lingcod spawning, studied rockfish abundance in relation to Howe Sound Rockfish Conservation Areas, and have successfully reintroduced black rockfish to Howe Sound. The Howe Sound group is led by Dr. Jeff Marliave, Vice President of Marine Science at the Vancouver Aquarium. He has produced over 85 scientific and technical reports and has conducted research in Howe Sound for 40 years. Dr. Jeff Marliave.
We're going to start tonight with an introduction to taxonomy, the naming of species, by Donna Gibbs, our diving taxonomist, and then Jessica Schultz will follow with a discussion of marine ecology in Howe Sound. At the end of that, I will give an example of one unique living habitat in Howe Sound, our very shallow glass sponge reefs. So we'll start now with Donna Gibbs.
Hello. My job as a taxonomist, I get to go diving. They pay me to dive. And when I'm diving, I get to keep track of all the plants and animals that I see on a dive. And what's been happening over time is we've built a really great long-term natural history database that gives us a basis for comparison. Now, why do we care about that? This animal here is a sunflower sea star. And recently, all the sea stars died. And so that we can go back on our data and see how they lived. We used to take them for granted. They were everywhere all the time. But now we can go back and look at them in more detail when they were here. And now that they're gone, we have to go back. Another thing is we have an algae that has been in Howe Sound before, but we're seeing it in greater numbers in different places. And so that's a change. And whenever there's a change, we're really interested in that. So we go back and see what we had before and pay attention to what's happening now. So that's what we do as taxonomists. The process of naming organisms is called taxonomy. And the important thing about taxonomy is it gives us comparison to other animals as well. So the more we know about organisms, the more we can compare them and look at them and see how they interact together. Now, we're going to talk a little bit more about taxonomy. Jeff has some ideas about this and how he wants to speak about it.
So I just want to make one point, which has to do with the fact that most of you are familiar with your family tree, how you're related to your cousins, your aunts, and your uncles. And you've probably seen a family tree of life, the relationships of different types of plants and animals to each other. I just want to give you a little reminder that all the ideas that get presented to you come from human minds. They don't necessarily represent reality. And so we're showing you a whole grove of family trees because, for example, the arthropods, which include the insects and the crustaceans, are considered, on the part of all the scientists at the British Museum, to not be from the same origins. They say that crustaceans are polyphyletic, meaning it's a grove of family trees, and they arose separately, different points in time, and came to the same evolutionary solution to problems in living. They got to have exoskeletons, so similar compound eyes, that sort of thing. And I'm a fish scientist, and that nice landscape tree way over on the right, chordates, represents animals' backbones. That's the fish. And in fact, my understanding is that, in fact, eels were never related to other types of fish, and the lungfishes are not related to our perciform fishes. The perch-like fishes didn't really arise from the soft-rayed herrings and salmon. They're unrelated. And Codfishes are an excellent example because their anatomy is very, very different. Their circulatory anatomy, their skeletal anatomy, they're just very different. They arose from separate ancestors. So fishes are polyphyletic as opposed to monophyletic, which means one family tree. It's just a different point of view, and there have been very forceful presentations of arguments that we all arose from the same cell. Got that? It's just difference of opinion, so keep your minds open, okay? Here, Donna.
Okay. Thanks, Jeff. It's not always easy out there. We can't just pick and choose when we dive, or we wouldn't have any reliable data. So we're out there in the rain and the snow, and we're taking quadrats and cameras, and we're just doing everything we can all year round. So there's always challenges to working in the marine environment. And some days, we're really lucky it's crystal clear to do our work, and we can see what we're doing and do multitasking, and this diver is taking taxon notes. You can see the slate, and they're just getting the data they need from the dive. But other days, this is what we're dealing with. On this dive, we had 20 minutes to go from the surface down to the bottom, find a bioherm that we'd never seen before. It's a little bit of a needle in a haystack because we throw the anchor, we have a pretty good idea where we are, but until we go down that line and find the bottom and look around, we're really not sure. So this is what we land on sometimes. This is about 90 feet in the darkness. But we make the best of it because on every dive we do, we try to get some kind of taxonomy, even if we're doing other purposes. And this case, it was actually looking for a bioherm. But because we may only get to this dive in our careers 3 or 4 times, we try to gather all the data we can because it may be the only data we get. So, find the fish. That's the fish that we just recorded on our way by. We saw it, and so that goes in the taxonomic database, even though it's not really part of the dive. So 20 minutes, surface, down, and back up to do this. Plus, Jeff will explain, he's going to show you a film of the bioherm that we actually found that day. So you'll get an idea of what we're doing in 20 minutes. To be a good taxonomist, you have to look for color and texture and shape, and that'll help you identify animals. So up in the top left, we have a couple of worms that look a little bit similar, but they're different stripings on them, and they're actually different colors. So that's what you'd have to look for in order to identify them. And there wouldn't be another worm that had striping like this, but there's something very similar in a couple of worms. In this case over here, we have 2 animals. They're limpets, and they're about the same depth, but they're very different looking, and you just develop an eye for it. So if you can pick out the color and the shape, and this little mark on this guy and the ridges on this one has a hole in the top. But if you're just swimming over them quickly, you might not notice those details. So the more details you get to know in your mind, know the animals, you get very good at picking out the different species while you're underwater. These animals here look very similar. This one is a sponge. This one is a completely different animal and a different phyla. This is a tunicate. This is a mushroom tunicate. And so the textures are very similar, but the animals are very different. So the more you learn, the more you'll do well with taxonomy. Find the species. This is a rock with quite a few species on it in a lot of different phyla. And this is what we do in a taxon dive. We don't always have time. This is about 5 minutes of a 40-minute dive because we're at shallower depths this time. But we have to, as we're swimming along, just calculate the abundance and the number of species, and we record it later. So in this one in particular, there are 17 species in 8 different groups. So we've got cnidarians. These are zoanthids. We have, I'm not going to go through them all, but I'll show you. These are rock scallops here. And on the rock scallops, there are sponges growing in the shell. We've got a fish here, a goby. We've got encrusting red algae. We've got encrusting coralline algae. We've got other varieties of red algae. Sponges, that's the sponge we just looked at, and so forth. And these are bryozoan moss animals. So there's a lot to see and a lot to record in a short time. But why does it matter? Well, it matters a lot because this graph here will tell you what has come of science data from citizen science over time. And Jeff is going to explain this. Oh, my God.
Now, you've probably recently heard this popular concept of the Salish Sea, Puget Sound in the Strait of Georgia. It makes good sense from a socioeconomic standpoint. There are a lot of cultural reasons to talk that way. But when looking at nearly half a century of seabed biodiversity data, natural groups fall out, and the purple color shows coast of Washington, Oregon, outer coast, fully exposed to open ocean weather. And the animals on that part of the American coast are quite different from up in northern British Columbia and Alaska, where the water's quite a bit colder. You just have a different group of species. And the west coast of Vancouver Island is somewhere in between. Now, coming around, Johnstone Strait at the north end of the Strait of Georgia has terrific tidal currents, but no exposure really to wave action and heavy storms. And that's characterized by a unique set of species, lots of interesting seaweeds and such, different from the community in the Strait of Georgia. Notice that this outer coast community in the Strait of Juan de Fuca goes all the way to Whidbey Island and the southern coast of San Juan Island. So you have totally open coast animals like purple sea urchins, surf grass, right in this area. And the species are so much an outer coast community that they act as a buffer, a big barrier between the Strait of Georgia community and Puget Sound. So Puget Sound, in fact, has a very special species diversity that's quite different from the Strait of Georgia. They're not the same, and this depiction of zoogeographic zones kind of blows the Salish Sea idea out of the water as far as seabed biodiversity. So Donna's going to continue.
And another reason it might matter is being able to identify animals can really help scientists. This is a picture of a fish that was seen at White Cliff Park. You can't always see the whole animal. This is the fish here. There's a tail and the back end, just a little bit of the body. It's a black rockfish, and this is a very important animal because of the releases that were done at Point Atkinson. This shows that the animals had actually reproduced and traveled as far as the White Cliff area. So knowing that and being able to help with any identification or animal movement is really good for science as well. So anything any diver can do for that is really great. So if you're a diver, and beware, because citizen science will get you. It's a lot of fun. You get in the water, you start to learn taxonomy just because it's interesting. And scuba snacks help you get started because there's lots of different shapes and sizes in there to give you an idea. So you usually start by just diving on your own and seeing things, and then you want to know more. So you get out the guidebooks and you come to people like us who are doing a lot of taxonomy as professionals, and we have our protege here, Laura. And then all the people around are a group of divers who were really good to us this year, and every year they helped us with the Ling Cod egg mass survey, which is a survey that we do every year to try and help us understand their reproduction and their egg sizes and how they're changing out there. And also we have rockfish abundance surveys running this time of year, and the divers are helping us do that too. So the more these divers can learn about the animals associated with these species, it's really helpful as well. So first, you learn to look in little cracks and look for animals like this that live in a crack. It's a clam kind of thing. And there's some worms over here. You can barely see them. So you got to get your light in there, and you got to get curious about what's happening. But if you're lucky, the animals will come to you as well. So an octopus might sneak up on you while you get your head in that crack, and just you turn around, and there it is. So they're curious. So you got to look up and look around sometimes, as well as looking in the cracks. Now, anyone can identify a Lingcod after a few dives because they're big, obvious fish. But the serious people will want to know what are these things on the fish as well. So you'll see more. So you always want to look closer. These are copepods. There's a close-up of what they look like, a parasite on the fish. And then we have associations. That's another way to learn taxonomy is here we have a sponge which has settled on the shell of a hermit crab and dissolved it. The hermit crab is fine. It doesn't hurt its exoskeleton, which is its outer coating. The hermit crab and sponge live together. And then the hermit crab is busy eating a little bit of stuff on the kelp. And in the meantime, the nudibranch has decided the sponge is a really good snack because they really like to eat it. So clearly the hermit crab has stopped long enough to let the nudibranch eat, but it won't last for long. But if you know these associations, you might figure out what's happening. And it helps you to look further for what might be there. Animal behavior is another part of taxonomy. It affects behavior, reproduction. There's the Lingcod that I was talking about with the Lingcod egg mass survey. There's going to be more talk about that. But the Lingcod will stay with its eggs, so you can be sure to find them staying in their territory at certain months of the years. Animal location is really important. This animal, and this animal are all the same. And in some locations, they look very different to each other. These ones they're grouped, and they're dark pink, and they're on walls. And in Howe Sound, they're singular animals out in the mud. You wouldn't think they were the same, but they are. But this one here looks similar. It's different. It has, if you could see close-ups of the detail on these tentacles, this one has some marks that this one does not. And you have to be careful not to just quickly identify that as one of these. And that's how you lead to new species. This animal was found in Agamemnon Channel about 80 feet about 10 years ago, and we still don't know what it is. But these ones here are all really common, and they're all the same. Another way to learn identification of animals is to learn about their life stages. Some of them look very different from the adult. So this one will grow up to be a China rockfish, but will grow up to look like this. This is the adult stage. And these two baby crabs are red rock crabs, and they have different colorings. They're not the same. They're the same animal with different colors, but they will all be this in the end. Is it nocturnal or diurnal? It's hard. If animals are out at night, sometimes they really are just nocturnal, so you won't see them in the daytime. So you have to dive at all times of the night and day as well to get to learn all the animals. It doesn't mean you won't see them in the daytime, but they're mostly out at night. Current. Around here we have the advantage of a lot of areas like Howe Sound, which are lower current than the outer coast. So we get distinctive animals in lower current areas, and so if you can tell that this animal here is absolutely high current, and you would never see it in Howe Sound. And then if you did, you need to tell somebody because it's a really big deal. It shouldn't be there. Something's changing, and again. So high current and low current will help you learn identification as well. So keep good track of your taxon, and get back to me on it, because I'm really curious if you have some lists for me and I can add to my database. Thank you.
Thank you. Hello, so I'm Jessica, as Jeff mentioned, and I'm going to talk a little bit about marine ecology, particularly in Howe Sound here. So as you know, the ocean is an absolutely fantastic place, and it's got a lot of unexplored living space within it. Most of the living space on Earth is in fact in the ocean, but we know very little about the ocean. So whereas on land, you can spend a lot where you can spend a lot of time investigating animals and looking at different habitats, in the marine realm, it's more difficult to explore because we're relying on technology and on the limited amount of bottom time that you have as a diver to look at different things. So one of the most diverse groups of species on the planet is the insects. There's over 350,000 species of beetles alone. So if you compare that to the marine realm, where there's only about 250,000 of all different types of species known, it tells us that there really is a whole lot to learn about the ocean. But you don't have to go down to the bottom of the Challenger deep in order to discover new species. So for example, this shrimp here, called Libius mundus, was only very recently discovered, about 15 years ago. And what's remarkable is that it's actually not that rare. You see it quite frequently. It lives in about 60 feet of water, so it's not particularly inaccessible to divers, but it can be easily confused with a lot of shrimps that you see more commonly. So unless you know really what you're looking for in the details of taxonomy, you might swim by and miss something like this. Now, our celebrity taxonomist, Donna Gibbs, actually helped discover and collect the first specimen of this animal. And so her cat, Mundy, is actually named after it, because Libius mundus means elegant cleaner. And as you know, cats like to bask in their glory as they clean themselves. So there's a fun anecdote there. Now, once you know what you're looking at and you know the species that are there, then you can start to ask other questions, like ecological questions. So ecology is the study of the interaction between different animals and plants and their environment. So we're asking questions like, why is there so much kelp in this picture? Is it because of the availability of light? Why, for example, isn't it all being eaten by the herbivores like the snails and the limpets and the hermit crabs? And will those populations of herbivores and plant eaters grow infinitely until they run out of kelp? Or are there other things that are keeping those numbers down? So those are the kinds of things that we're interested in asking. And those things can get a little bit complex when you start looking at all the different types of variables that are interacting in any one environment. And so in an effort to get our heads around what's going on, ecologists divide ecological factors into two main groups. The first one is the abiotic factors. So an abiotic factor is anything physical, any physical characteristic of the environment, such as the light, which we mentioned earlier, or the chemical composition of the water, things like temperature. The other category, biotic factors, is the factors that involve other living organisms interacting with one another, such as predation or herbivory. So we'll go through a few examples of both of these. With abiotic factors, one of the more obvious factors is light. So light can limit or impact organisms in a number of different ways. Here in this picture, we see an anemone that looks green. And the reason it's green is because there's microscopic algae that actually live inside of its tissues and photosynthesize. And they can only exist where there's enough light for them to produce energy out of the sunlight. So this same species of anemone, if you find it in deeper water, will actually be white because that algae can no longer exist. Another interesting thing that happens in the marine environment is light attenuates or gets absorbed with depth. So this canary rockfish looks orange here because this picture is taken in an aquarium. But if you were actually at depth, it would look brown. And there's other things going on, like we have some more photosynthetic pink algae and other animals that could be limited by light as well. Our next example is temperature. So most marine invertebrates and fish are exothermic, which means they don't regulate their internal body temperature like we do. So when the environment gets colder or warmer, the animal will also get colder or warmer. So that has a lot of impacts on physiology. Metabolism slows down in cold water and a number of other physiological functions as well. And so because of those physiological differences with temperature, it can really change the composition of an environment. So this is a sand buoy. It's an oceanographic buoy that measures abiotic variables like salinity, oxygen content, temperature. And it's placed here in an area where there's cold water upwelling. And a nearby reef where there isn't cold water upwelling can have an extremely different ensemble of organisms living, even though all other factors are very similar. So temperature is very important. Another one is salinity. So that means how salty the water is. And if you've ever spent a lot of time in the bathtub and your fingers get wrinkly, that's because the water in your body is moving outside in order to compensate for the salinity. It's called osmoregulation. And for marine animals that are in the bathtub, so to speak, all the time, osmoregulation is a constant consideration for them. Osmoregulation takes quite a bit of energy, and so having different salinities can really influence where organisms are able to exist. Similarly, oxygen also will impact where organisms live. You can also have other chemical elements of the water, like in this case, silica will affect different organisms. These are diatoms. They're microscopic photosynthetic algae, and they're made out of glass, essentially. And glass is silica. So they'll do better in areas where there's a lot of silica content in the water. Another one, pH, that refers to how acidic or basic the water is. And acidity is a widely studied variable at the moment because of climate change. When the atmosphere gets higher contents of carbon dioxide, it gets absorbed into the ocean, which decreases the pH of the water, making it more acidic. That process is called ocean acidification. It's been referred to as the evil twin of climate change. And for a lot of organisms in the marine realm, that makes a big impact. So this snail here builds its shell out of calcium carbonate, which is limestone. And building limestone is a lot more difficult in acidic water. Same thing with the acorn barnacles here. So any animal that builds a shell out of calcium carbonate may have a hard time with climate change. And so this is a really active area of research at the moment. Finally, I'll just mention quickly water movement. Donna already mentioned how low and high current can have a different group of organisms living in them. And that's because water current and water movements will affect all the other variables, such as oxygen, salinity, temperature, and to some degree, light will also change with current as well. And when you consider all abiotics conditions together and over long periods of time, that's when you start thinking about climate, which is the long-term changes in different weather conditions. So now we're just going to move on to the biotic interactions. So the first one we'll talk about is predation and herbivory. So that's animals eating other animals or animals eating plants, in this case algae. And that's one of the more readily observable interactions you can see in nature. Biotic interactions can be considered either positive or negative. So in this example, for the sea star, which eats the sea urchin, it's a positive thing for the sea star because it's gaining food. And of course, it's a negative thing for the sea urchin because they're threatened by predation. And exactly the same interaction cycle is taking place between the plant eater and the algae. The next example is competition. So in this picture, we have a snail and a chitin, which both graze on the surface of rocks for microscopic algae or larger algae if they can get a hold of it as well. And so they're competing not only for food, but also for space. And in the case of competition, it's a negative interaction because both organisms have to lose out in order to compete for the resource. They're spending more energy to compete, or they're obtaining less of whatever resource you might be referring to. So competition isn't necessarily for one thing, but it can be for a number of different things at the same time. Next, we'll just mention parasitism. So here we've got a shrimp that's got a parasitic isopod that's actually laid its egg mass underneath the exoskeleton of the shrimp. So in this type of interaction, it's good for the parasite and negative for the host. And while most parasites don't kill the host because it's actually better for a parasite if the host can persist because it can draw the benefit for a longer period of time, there's still going to be at least some negative impact for the shrimp. It's going to have to sustain the injury of having its exoskeleton ruptured, and it's going to be more vulnerable to infection and that type of thing. Whereas for the isopod, it gets protection for the eggs. And in this case, once these eggs hatch, they'll rupture the exoskeleton, go off to infect a new host, and the shrimp will not die. So parasitism can lead to other interactions as well. So Donna mentioned before that the Lingcod can have parasitic copepods underneath the skin. But in this picture, we have our cleaner shrimp, our elegant cleaner shrimp that we mentioned earlier, down there picking off those isopods as a form of food. So this type of interaction is actually called mutualism or commensalism, which is our next interaction that we're going to talk about. In this picture, we've got a nudibranch, and it's got a scale worm that lives on the side of it. It's not harming the nudibranch or inside the nudibranch, but just living there as a way of transportation and protection, potentially. So these stinging projections on the nudibranch might be protecting the worm, and the worm will actually rear up kind of like a guard dog when other organisms get closer. So that's been observed when this worm's on snails and limpets and such. So it's probably going to happen with the nudibranch as well. So in this type of interaction, it's positive, or neutral for both animals. So that one's mutualism. When biotic and abiotic interactions are present for a long period of time, they can lead to population changes. So one example is with the Sixgill shark. It hadn't been seen in our biodiversity records previously at all until 2011, when it was seen once. It was seen once in 2012, and it's been seen 3 times this summer. So that might be from a result of more food availability with the return of potentially the pollock and herring and other animals. We've also been seeing more cetaceans in Howe Sound recently. And that might be from a whole number of different factors which are probably related. We can also look at more than one interaction at the same time. So ecologists often talk about trophic levels. So trophic levels are defined by how an animal or plant obtains energy. So in the primary layer at the primary producers, they're obtaining energy from either the sun or from chemistry. They don't need to consume anything else to get energy. The second level, the herbivores or plant eaters, are consuming the primary producers. And then the carnivore or predator will be consuming the herbivores. And you can have multiple trophic levels in each division. Trophic levels can regulate populations. So ecologists like to talk about either bottom-up controls or top-down controls. So that refers to what's limiting the growth of a population. In the marine world, there's not very many examples of bottom-up control, but a terrestrial example might be a hare and a wolf. So when the hare population is reduced, the wolf can't grow as much in numbers because it doesn't have a food source. So that's a bottom-up control. One example that we do have in the marine realm is that of plankton blooms. So when there's more sunlight and more nutrients available, which is on the bottom level of a trophic level, then the microscopic organisms are able to reproduce more rapidly. And this is a dinoflagellate bloom in Howe Sound. Top-down mechanisms are more common in the marine realm. So this is where a predator or herbivore is limiting the level below. So this is a famous example in ecology where the presence of a sea otter is reducing the number of green urchins. And in this case, purple urchins. And similarly, the purple urchin reduces the amount of kelp that can be present in the system. So when the sea otters aren't around, the urchins can just eat unchecked and consume a lot of algae, which will bring down the kelp forest, and that has a lot of other ecological impacts as well. So then we can have indirect interactions. When sea otters are present, they limit the number of urchins that are around and indirectly increase the amount of kelp. So that's why sea otters are such an important element of the ecosystem, because they can maintain kelp forests, which allow for juvenile fish to have more habitat and other organisms as well. Kelp systems are very biodiverse, so this is a really highly studied system. And when you get these indirect interactions like this, it's referred to as a trophic cascade. So that's where a change at one level will influence another level. You can also have food webs where there's interactions happening in more than one direction. So if you're just looking at the top-down or bottom-up direction here, we've got the ochre sea star, which consumes all the other organisms in this food web, Nucella, which is a dog whelp, and both of these animals will consume mussels and barnacles, and then the sea star will also eat the chitons, limpets, and the gooseneck barnacles as well. So that's when you're looking up and down. But from side to side, all the organisms in the lower level are also competing with one another, so they have a negative interaction between the different groups. And that can lead to some more interesting types of interactions. The sea stars primarily like to eat the mussels. And so when you look at the intertidal realm, you can see that the sea stars will limit the number of mussels that are around. And by limiting the number of mussels, actually is a benefit for the other animals in the bottom of the food web here, because they compete for space. So without the mussels there, the other organisms are able to reproduce more, occupy more space, and actually by having the sea stars present, increases the biodiversity in the system. And when you have that type of interaction, it's referred to as keystone predation. So the sea stars are having a disproportionately large impact on the ecosystem by increasing biodiversity when they eat mussels. Does keystone predation occur in the subtidal? The subtidal ecosystems are a lot less studied, but we think that the sunflower star here might be having a disproportionately large impact on the subtidal community. It's a voracious predator. It eats pretty much anything that it'll come across, including snails, urchins, chitons. Can't see what that is. I think it's a limpet, hermit crabs, and possibly even kelp. And not only do these animals get reduced in number when they're eaten by sunflower stars, but they also have adverse behavioral reactions by moving their distributions and expending energy in order to get away from the sunflower stars. So last year, since about 2010, sunflower stars have been extremely abundant in this area. However, last year, about this time, in late August and early September, the area experienced a sea star wasting syndrome. So there was a mass mortality event of sea stars, and to date it's been documented from Alaska down to Southern California. So in the sea star wasting syndrome, the sea stars would lose their internal body pressure. They would start twisting, developing lesions, and eventually disintegrate in place. So you can see in this image here that on the left and right is actually a river of ossicles, if you like, which is the decaying necrotic debris from all the sea stars. This picture here is the same rock as the previous picture where all the sea stars were on there. So it provided a natural experiment to investigate ecology. We're not sure what's causing the sea star wasting syndrome, but it's likely a combination of biologic factors like their previously abundant population and abiotic things such as climate variables. So that's still under investigation. And one way that we're investigating it is by looking at the ecological impacts. So here we're doing a underwater transect. So that's essentially a measuring tape that's laid down underwater. And we'll go down and we'll count all the fish, invertebrates, and algae along that length of measuring tape. And in 2009 and 2010, a previous study did the same thing, looking at about 40 different sites in Howe Sound. So we're going back to those sites this year and next year probably as well. And we'll do the same study at the same sites and the same depths so that we can compare the community before and after sea star wasting syndrome. I don't have too many results to share just yet, but one of the most striking things we've been seeing is a huge increase in abundance of the green sea urchin, probably because it's been released from predation by the sea stars. And this might have other trickle-down effects in other parts of the community too. So we're getting to see some of these concepts in action, like trophic cascades. So the trophic cascade in this case is that the sea urchins consume the agarum, which is the sea colander kelp, and the spot prawn actually uses the kelp as somewhere to settle when it's a juvenile. So the spot prawns will settle out of the seawater and use the blades of the kelp as shelter, and they'll live in and on the kelp. But with the urchins being so abundant, they're consuming a lot of the kelp. So here we have what's sort of the normal or pre-sea star trophic interactions. So we've got the sea star consuming the sea urchin consuming the algae, and the algae providing a positive benefit for the spot prawn by supplying nursery habitat. And the indirect interactions are that the presence of the sea star is good for the agarum because it reduces the amount of herbivory by the sea urchins, and it's also good for the spot prawn because it allows more habitat for nursery grounds. But in the absence of the sea star, we have an abundance of the sea urchin, and now there's even a reduced amount of kelp and a reduced amount of habitat for the spot prawn. So here we get to see a local example of trophic cascades and keystone predation having an impact here in Howe Sound. So that's an active area of research for us. And with that, I'm going to pass it back over to Jeff, who's going to discuss unique habitat here in Howe Sound.
Thank you. I'm going to switch gears now and talk about just one thing because it's kind of unique in Howe Sound in that we can actually do a lot of diving around glass sponges. And this is a shot of a cloud sponge, Aphroclistes vastus, and you can see lots of little squat lobsters. They're a type of crustacean, and they can be super abundant on cloud sponge reefs. And this is something that is really special to be able to see because in most parts of British Columbia, you have to have a submarine to get deep enough. Now, sponges are very primitive living things that have a structure that enables the tissues to pump seawater through, and they filter out food items through an opening called an osculum. And in the glass sponges, the skeleton or spicule network, the architecture of the sponge, is made up of glass, of silica, just like Jessica was mentioning, silica being the building block for diatoms, part of the plankton that blooms in the ocean. We and most of the plants and animals on Earth are made of carbon atoms. Carbon is the building block for most life, so it's kind of special to have these silica-based organisms. Now, there are a couple different types of glass sponges, and the chimney or boot sponges are a type that I like to say functions like a reverse vacuum bag because the glass spicules are in a sort of a sloppy mat on the outside, and during a diatom bloom, they build up, they filter water through, and a big mat of diatoms builds up, and it looks very, very dark brown. And then over the weeks and months, it gets slimy and dusty looking, and so it's breaking down. Now, the sponges directly feed on bacteria, and bacteria are what's breaking down the diatoms, but the breaking down of all these diatoms may actually release a higher concentration of silica into the water. Nobody really understands what concentration of silica means to growth in glass sponges, but it's something that that we can take an interest in Howe Sound. Oh, what have I done? Come on. Oops. So there used to be sort of a legend that these glass sponges are centuries old, don't touch them, and we've been following them in Howe Sound for quite a while, and we've found with the boot sponges that we can actually follow the whole life cycle. We physically tagged some of them, but we've had real trouble in that they change shape so much that you can't really recognize them from one season to the next. What we found is that we can recognize the geology of the rock formation. We kind of map the crevices and ledges on a cliff and keep track of the sponges. And what happens is they get sort of disorganized. They get all ripply and rumply and form new oscula. This is probably several boot sponges clustered here. But eventually, their tissues get so disorganized that they just fall apart. And this occurs from young to old over the space of about a decade. So this is a young glass sponge with one tight osculum. And the genera and species, getting back to taxonomy, Rhabdoclyptus dawsoni and Staroclyptus dowlingi, were erected back in the 1890s when scientists dissolved the sponges in acid, looked under a microscope at the shape of the spicules and saw the different shape of spicules. So they actually came with two different genera, Rhabdoclyptus and Staroclyptus. Staroclyptus being all flared out, sort of like the Arceae victrola trumpet. And what I suspect is that, in fact, if some young scientist were to go back, good old-fashioned scientists dissolving these sponges in acid and looking at the spicules, something that people aren't really doing right now, you might find that when they get all rippled and disorganized, that their spicules are those of the Staroclyptus dowlingi. It could be that these two different genera are really just life stages of one type of animal. So that's one of the advantages of being able to more or less live with them in Howe Sound and spend a lot of time looking at them. So the cloud sponges are a different family within the glass sponges, and we only have two genera in Howe Sound, Aphroclystes vastus and Heterocone calyx, and for the most part, all we see is Aphroclystes, and it's the one that's named the cloud sponge. And they occur from way up in Alaska all the way down to Southern California and such, living on rocks, usually at tremendous depths. It was only more recently, though, that they were found to form reefs in British Columbia. Oops, get that wrong. Now, this is a cloud sponge in very shallow water in Howe Sound that it was about the size of a Volkswagen, and it had been a big, beautiful sponge up until 2009, when we were actually diving on it at the beginning of the great 2009-2010 El Nino, a very serious warming period. And the whole thing turned translucent and looked pasty, and Donna was noticing a particular type of starfish that's a predator on sponges, zooming in, if a starfish can zoom. But their arms were pointed as if they were making a rapid high-board dive onto these, and they were actually climbing onto them, meaning that the tissues were failing, and we thought the whole thing had died because it was so shallow. And this is about a year later, a little over a year later, and the dark brown is where most of it is dead. And then around the edges, some of it seemed to survive and recover and actually started growing fairly rapidly because 2010, 2011, we had a La Nina, a cold period. So this is the beginning of the cold period. And please note, there's a juvenile quillback rockfish right there. There's one here. There's one peeking over the top here. And there's one up right here. And so these sponges are a very important nursery for juvenile quillback rockfish, a species that's listed as threatened. Now, if the cloud sponges are growing on rocks, on a cliff like this rather well-known cliff at Texada Island, it's called a sponge garden. And lots of baby quillback and yellow eye rockfish will settle into the area around the sponges. They hide inside the cloud sponges, and then they're out on the rock community finding all kinds of tiny little animals that they can feed upon. If the cloud sponges are growing on dead bodies of other cloud sponges, previous cloud sponges, then that is a sponge reef or bioherm. So glass sponge reefs are built up on silted-in skeletons of previous glass sponges, and those silted-in skeletons become geologically stable. So the geology of mountains, including the Pyrenees and the Alps, includes this chalk-like substrate that was once glass sponge reef. And in ancient times, the largest living landform in the history of the Earth was a glass sponge reef that spread all across Europe. And it was thought that they had all gone extinct during the Jurassic until just a couple decades ago when they were found in British Columbia. And the sponge reefs only occur in British Columbia. And actually, this picture here, this photo mural, is a collage of snapshots that Donna and I took in Howe Sound on a reef that's shallow enough to dive on. And in all of British Columbia, the only place we know where you can actually dive on these sponge reefs is in Howe Sound. So this is what it's like. This is a dive that Jessica and Donna took on a sponge reef at the entrance to Howe Sound. And the second diver, Donna, was providing the lighting. And it makes it a little more spooky. No bright lights right at the camera, but it gives you a feeling for what it's like. This was in broad daylight, but no light gets down to the reef because of the plankton bloom. And you can see these broodstock quillback rockfish that have already given birth by this time in the season, but good numbers of them taking refuge in the cloud sponge reef. And Donna was talking earlier about I want you to know, it was an English sole that they found on their way to the bioherm. And when they get there, they still don't really know how big the glass sponge reef is, what its extent is, how far it goes. And that's part of the mystery of diving on these glass sponge reefs. But they're very convoluted. And so where you would have had just smooth glaciated rock, you have this very complicated structure that provides a lot of refuge for certain types of animals, which include rockfishes and the squat lobsters you saw in an earlier photograph, any number of crustaceans. But it's only possible to dive on compressed air in Howe Sound and see this type of reef structure. So it's something that we have specially available to us, and we're very, very pleased to be able to do that. Now, looking at the seabed biodiversity records that Donna has amassed through various dives in areas with sponge gardens and bioherms, it actually turns out that where the baby rockfish settle and grow in their nursery stages on the sponge gardens, which are the blue bars, have a huge biodiversity. And that's, for the most part, because of all the different types of plants and animals that live on the rocks around the glass sponges. Whereas on the bioherm or the sponge reef, really you have fish and you have shrimps and crabs on the sponges. And you got sponges, porifera. And there's one type of worm that lives on the mud at the base of these sponges. And there's one type of nudibranch or sea slug that lives similarly at the base of the living part of the bioherm. But it actually has much less biodiversity. And so it's actually not good nursery habitat. What it is a good grow out for rockfish. And it's when a quillback rockfish gets big enough to eat those squat lobsters, then they move on to the bioherm. And very typically there are sponge gardens immediately adjacent to sponge reefs. So it's very important as habitat. I want you to take note that this growing quillback sticking inside a sponge is causing a bit of physical damage to the cloud sponge. They're very delicate growths. And the squat lobsters, of course, are very important food for the quillback. We don't know if some of these shrimp get eaten by rockfish. There's one type of shrimp that is not really named, and it's a sponge reef shrimp. It doesn't live anywhere else. So there are still lots of things we have to figure out. Now, what's so special about being able to dive on a bioherm? What's the big deal? Well, they're mostly in very deep water in British Columbia, so you have to send down a remotely operated vehicle, which means a little mini submarine with a video camera in it, tethered to a ship where an operator is operating it like a video game. And we think that divers can be a little less clumsy than a remote vehicle, which can cause damage. And so that's one of the beauties of being able to study them in Howe Sound, that we're not doing this level of damage. This is Pharia aca, by the way, a type of very delicate cloud sponge that does not live in BC, and that's a vase sponge in that last shot. So that's kind of a cruel comparison showing that footage, but fishing gear causes a great deal of damage to cloud sponges. My cocktail party story is that, these cloud sponges have the same structural rigidity as a meringue fresh out of the oven. You flick it and it's going to shatter. So in May of 2012, we were very upset. That same big sponge that had been hit by the El Nino and that was recovering got sliced. It just got sliced in half by a downrigger line. And over on the left, this is the slice. This just like a cheese wire, cutting it through, and you can see the internal structure. And this part had folded down. I was worried that it was going to become dislodged, but in fact, by the end of the year, it was still lying there, and the cut open parts were healing over. There's no glass spicule architecture inside these narrow healed spots that can have a sort of a spiderweb lacy appearance. They can actually open and close in the space of a day. But this is La Nina weather here. It's very cold, and there was tissue repair going on. Now, at the time of this incident, we were upset to see it. We were really upset. And we wanted to do a blog posting about it. We were told, no, you have to have something on display, if we're going to talk about it. So we went back, and we were going to bag one of the fragments. And over on the upper right, this is a piece that's where it was cut off, and it's lying against a full living cloud sponge down in deeper water, and we couldn't get the bag over it. I was stretching the bag, and Donna was looking at it, and so she hesitated, took out her dive knife, and made a big slice parallel to this slice, and all of the good outer morphology on this side went on display, and we posted our blog narrative about the damage that can be done. But at the time that we got the piece bagged, I looked at this giant Texas toast slice of cut cloud sponge, and my heart was breaking over it. What can we do? What can we do? And so big, I gently picked it up, looked around, and there were two big rocks at right angles on a downhill slope, and I sort of tucked it in on the uphill side and thought, that'll be good. We went back and saw, it had wafted away, and so I got on my knees and I walked a couple more rocks over, and as soon as I would touch the sponge, it would start shattering, so I just got it as close as I could without quite touching so that it was stuck in place, had about 1 millimeter of wiggle room. And so that's May of 2012, and this is December, and this is part of one of the slices. And you can see that it's healing over everywhere it was sliced, and it's getting better. And this is a break that's healing over. And so we were very interested in that. And at this point in time, 2 years later, I'm going to take you for a little tour of this fragment. Actually, it's a series of fragments because there are a couple of loose pieces that were being crushed in my hands, and I tucked them down in a loose spot. And from going clockwise, this is the view from the west, and you can actually see where it's touching the rock, and like buttress roots from a tree, it is fused. Everywhere it's touching, it is fused to the rock. And then this is, looking from the south, this is the biggest rock. And that's where I was looking down in that other shot where it was healing. And then from the east, these were loose pieces, and they have, here. I'm going clockwise. So this one right here, these were the loose pieces that have grown together. And it's now growing out from the rocks that I had placed. So these are the two rocks that were the big rocks. And it's now looking like a nice little cloud sponge. And this is getting to a view from the north. And you can see that it's sticking out from little rocks that I placed and bigger rocks. And so it's doing really well, but, yippee. We put it there, and it showed that it has the ability to reattach to rock, but in nature, the odds are probably vanishingly small that a shard would fall down into a perfect fit so it wouldn't wiggle around and wouldn't get battered. When we've seen fishing gear damage in other cases, we've just seen it all deteriorate, get swept away, and it dies quickly. The one exception, though, might be on these glass sponge reefs where all these arms are coming out, and I've failed to mention, this is all one cell. Like, it's so primitive. It doesn't have cell walls, so there's no tissue compatibility problem. And if a living arm gets broken off on a glass sponge reef and it comes to rest on other arms of living cloud sponge, it probably can fuse and continue to grow. And so we've seen a lot of evidence of breakage resulting in subsequent growth on the one reef that we've been studying. And it's very likely from what we see on a drop camera that where there's damage by fishing gear, extensive damage, these sponges struggle to fight back and earn a living even in the presence of humanity. So I want to talk about protecting these sponge reefs or bioherms. Now, I have actually made a presentation with those taxonomic data and such, and argued up in Alaska that since hook and line fishing is not allowed in rockfish conservation areas, these areas in orange, in order to protect rockfish, there should not be other deleterious impact on rockfishes in those rockfish conservation areas. And because it's not a hook and line gear, prawn trapping is allowed inside the rockfish conservation areas. So my argument was either the prawn fishery should adopt a code of practice so they will avoid sponge gardens and sponge reefs, or they should not be allowed inside rockfish conservation areas because they're damaging. Now, that's a really strenuous case to make because between 25 and 33% of British Columbia's known rock reef substrate is inside rockfish conservation areas, and that would be a serious economic damage to the fishery. The reefs themselves are not nearly as great a spatial area, and they could be avoided. Now, the pink dots represent known sponge reefs in Howe Sound. They're being found fairly rapidly by citizen science, a fellow who operates a drift drop camera. Right here, off this, there's a feature coming out off the northeast corner of Anvil Island. Just a few weeks ago, they discovered yet another sponge reef. None of these sponge reefs in this part of Howe Sound are encompassed in the measures that are coming up to protect sponge reefs in the Strait of Georgia in terms of federal government policy. These reefs down here are included. And the difference is that in the Strait of Georgia, which is a shallower slope to its seabed, multi-beam imaging could detect the presence of the sponge reefs. But on the steep pinnacle features in the walls in Howe Sound, the resolution wasn't high enough to reveal the presence of the sponge bioherms. So the indirect method used to indicate where they might be wasn't really adequate for Howe Sound. Now this has us concerned because we know these reefs are here. We know that, for example, off Lions Bay, there's a tremendous amount of prawn fishing, both commercial and sport. And so there's an active effort being made to convince people at the Lions Bay Marina to do their fishing near the shoreline and not on the offshore pinnacles. And that seems to be working with the sport people. There's a lot of negotiation going on with the government over these reefs down near Passage Island and White Cliff Park because the live fishery to Granville Island, that market depends critically on the prawn fleet being able to zoom out and get right back in quickly. So it's a really serious problem. And one of the obvious solutions that we think has to do with these diveable reefs. There are only 5 areas. This is one reef off Passage Island. That's the dark, spooky video that Jessica and Donna got. And then this is a diveable reef south of where the ferry's going to Bowen Island. And it starts at 125 feet. Thank you, we're not going to go there. That's really deep. This reef starts at about 80 feet. There's diveable reef that's really nice on a pinnacle off of Halkett Provincial Park. And it's a little deeper, it's over 100 feet. But this is a reef that starts at 125 feet, so we haven't been there. And then finally, up at the Defense Islands, another set of reefs, 2 reefs that start at only 80 or 90 feet. And so they're very amenable to study. And that's where we've been doing some work. And in fact, what have I done here? We've decided to go public and to not keep a secret of where the bioherms are, and to let the diving public know about them and actually encourage some citizen science. We would like to see Howe Sound become a world destination for diving on these glass sponge reefs. It's the only place you can do it. What I realized looking at videos taken between 2007 and 2013 is that just like with the boot sponges, their growth is so amorphous that you really can't identify the sponges from one year to the next just by looking at them. You have to know exactly where you are. So what we've done is, this is a cartoon of the sponge reef off the near Defense Island, and we've put in stakes with a coating so that we know where each of these stakes is. There are 1, 2, and 3 black stripes on this series of stakes, and 3, 2, and 1 green stripes, and then 1, 2, and 3 blue stripes. And we anchor the boat up on a rocky reef. This is with north at the top of the, this is the Defense Island. We anchor on the rocky reef here, and then this is the sponge reef. There's another sponge reef out offshore. We have a polypropylene line that we follow over sort of no man's land where it's spooky, muddy bottom, usually very dark. And then when we get there, we now have a temperature monitoring device there, and the original 2 black stakes pounded in the mud where there had been no sponges, but there are now sponges growing right around it, probably because we think that during the El Nino that this whole reef kind of got decked. Like there was a lot of collapse. Some of it tumbled downhill around those black stakes, and it seems to be regrowing now. We don't know where we were when we shot earlier videotape, but we can look at videotape from 2012 and say, wow, well, those are obviously more than 2 meters tall, and it's just not that way anymore. So we can't prove anything about what we were seeing then. Now we've got these stakes in, and we're inviting other people to go, and the idea is to go around those perimeter stakes and always keep your fins and your body away from the glass sponges. Don't do any damage, because just like fishing gear, if you swim right into them, they will shatter. And then always to have the color code of one of the stakes in the frame of your photograph or your video, and then we'll be able to analyze shots over seasons and years. So this is videotape of the 3 green stripes at the deepest part of this sponge reef. The frame is bouncing around because we've stabilized the video. There's a stripe at the bottom so we can see how deep the mud is, and eventually we expect these stakes to bury themselves as the bioherm builds up, collapses, gets sedimented, and then swimming over to the next stake with 2 stripes. We have a landmark in the frame, and we're getting an idea of the relative height of the sponges compared to the stake, and you get a sense of perspective looking in the distance. This happened to be full sun and clear seawater, so no need for scuba lights. We actually had our sense of orientation in this particular rare bit of video footage. Usually it doesn't have such good conditions, but down here you can see small sponges near the stake, so we'll be able to look at them in the future and gauge their rate of growth. And the camera panning down, keeping the stake in view, and then you see small sponges near the base of the stake, and we'll be able to gauge the rate of growth. And then this is at the greatest depth, the greatest distance from the center of the reef, where there's 1 stripe on the stake. With the full daylight, without a video light, you can't really see that the stripes are green, but when we're using lights, the colors are perfectly obvious to us. And you can see the matrix of dead sponge that's been silted in, and it's actually meters deep. You can't find the bottom of it, but it's pretty stable. And where we did see a lot of collapse, stuff obviously rolled downhill in one area, and we see new sponge growing up, and we think it's growing up fairly rapidly. So this contradicts some of the thought in the literature that said that they were extraordinarily slow growing. And it turns out the literature proving extremely slow growth in glass sponges was all published from work where the observations were made during El Nino years. And in fact, during the La Nina years, you seem to get very rapid growth. So climate regime shifts are things that we have to pay more attention to. And that's the end of that video. So thank you very much for your kind attention. End of show. Does our huge audience have questions for us?